Happy Saturday, scientists. Let's review. As you can tell by my bow tie, there's more biology today. We're tackling B2 called Scaling Up. As always, if you have any questions, please leave them in the comments below and we'll do our best to get to them. There's also a worksheet that you can use for some practice questions. Let's get started. In B1, we visited cells like those I'm sharing the screen with now, and learnt about some of their vital processes. If you need to revise that topic, there's a card on screen now. Today, we are going to look at the cell on a larger scale and see how the processes underpin the cell's function both on its own and as part of larger systems. The first thing the cell is going to need is to be supplied with nutrients. And to do this, we're going to need something that links in biology, chemistry, and physics, and uses a bit of underpinning theory from all of those. I think it's time for a home experiment. For those of you who love to cook, or who want to make your family happy, why not make them a nice fry-up on this Saturday morning? I'm obliged to say be careful, of course. If they aren't awake yet, you might find that soon they are. They're woken up by the delicious smell of your cooking. How did that happen? Well, it's diffusion, the random movement of particles from an area of higher concentration to an area of lower concentration. And that's our first topic today. As I mentioned, diffusion is the movement of particles from an area of higher concentration to an area of lower concentration. And it's really important that we understand these ER bits. There doesn't have to be a large difference between the two concentrations in order for diffusion to happen. And diffusion will be affected by a number of different factors. The first of these is temperature. And though in biological systems, especially systems like humans where the body temperature remains constant, this is less of a factor. It's still important to understand how this happens. And to understand it, we need to link in our chemistry knowledge. So it's very good, as always, when we're studying, to try and link together other subjects and to understand how the things that we've studied apply in different cases. With temperature, we know that particles will move faster at higher temperatures. They increase their movement as the temperature gets higher. If they're increasing their movement at higher temperatures, then when you have a higher temperature, you're going to have a higher rate of diffusion. And this is because your particles are going to be moving faster, and so this random movement of particles from higher to lower is going to take place faster. Next, we have the concentration gradient, and this is the difference in concentration between the two. If there's our cell, and inside the cell we have a few particles, but outside the cell we've got quite a lot of particles, well then the concentration gradient between those two, or the difference in concentration between those two, is quite high. Whereas in the case here, where we kind of have the same number of particles inside and out of the cell, then that difference in concentration gradient is nearly non-existent. Next we have the surface area, and this is a measure of the total surface of the cell. The higher the surface area, the higher the rate of diffusion. And there are special areas in your body and special cells in your body that are adapted to have a very high rate of diffusion, and therefore they are adapted to have a very high surface area. An example is the alveoli in your lungs, which are those little air sacs. Right, so they, by having this spherical shape, have quite a large surface area. There's also your intestines. If you were to look at a cross-section of the small intestine, it might look something like this. This would be the lumen, the open area in the middle, and then that would be the highly folded surface all around. And that allows the small intestine to have a high surface area and therefore quick diffusion. While we're on the subject of the small intestine and the alveoli, the next factor, distance, will come into play. If I were to draw the blood vessels in here, they're going to be right around the surface, and in fact there's going to be a network of blood vessels kind of all over the surface here, as close to that surface as possible. And that's because the higher the distance, the slower the diffusion, because the particles have further to travel. If your body is trying to increase the rate of diffusion, then it's going to want to make sure that those blood vessels are really, really close to the surface there, and that diffusion is able to happen rapidly. There is another particle movement that is very, very important in biological systems, and that is osmosis. This is again a passive movement, so energy is not required, but in this case it's water moving from an area of low particle concentration to an area of high particle concentration across a partially permeable membrane. Now, sometimes instead of writing low particle concentration, what we'll write is high water potential. And instead of high particle concentration, we might write low water potential. It is still moving down a concentration gradient, therefore it's passive, but in this case it's moving kind of down the concentration of water, which happens to be the reverse of the concentration of particles. So if you have your partially permeable membrane, or if it's the cell membrane, your selectively permeable membrane, and you have some particles here, nature wants for things to be in balance. But if these particles aren't able to go through the membrane, then what's going to happen is that water molecules from outside of the membrane will move in order to try and bring the two concentrations equal. And this can be very useful, for example, in the root hair cells of plants, where we can get the water to follow the minerals in. And getting those minerals in is a little bit of a challenge for the plants, because they're moving those minerals up the concentration gradient. And that means they need to use something called active transport. It's the active movement of particles, and so that means that energy is required. As we learned in B1, energy in terms of cells means we are dealing with ATP. So ATP is going to be required to move particles against their concentration gradient. They're going to want to move the other way. We're going to force them to move the way we want them to move. And we do that using something called a carrier protein. If this is my cell membrane, I'll have my special carrier protein in the membrane there, and then it'll have an active site that is the shape of the substrate that I'm trying to move in. So let's say I'm trying to move this substrate in, then there might be sort of a half circle on both sides to kind of grip that substrate. And then using some ATP, most carrier proteins are going to have a sort of flip that they do, and then they'll have that substrate moved into the cell. That's going to require energy. As I mentioned, it's something that's used in the root hair cells of plants. It's also used in your nerve cells in the sodium-potassium pump. Those are two applications for that. All of these transport types, but especially the passive ones, are going to depend on something called the surface area to volume ratio. And that's the ratio of the surface area around the cell, if you'll permit a very well-drawn spherical cell here, then the surface area of that cell compared to the volume of that cell. I'd like you to take a moment to think, would that be better with larger cells or better with smaller cells? Let's do a little bit of an example here with some numbers. Let's say that this cell has a radius of 1 centimeter, and we have another cell, and just to make this quite contrasting, this cell is going to have a radius of 10 centimeters. I'd like you to calculate the surface area to volume ratio of these cells. And that will mean that we need to know the formulas for that. We need to remember that the surface area of a sphere is calculated by using the formula 4 pi r squared, and that the volume of a sphere is calculated using the formula 4 over 3 pi r cubed. I'd like you to pause the video and to do those two calculations, and then to check and see if you got the correct answer. If we're doing this calculation here, we're doing 4 pi and then the radius was 1 centimeter squared, and that's going to give us a surface area of 12.5 centimeters squared. And if we do that with the volume, so we have 4 over 3 times pi multiplied by 1 cubed, and that will give us a volume of 4.18 centimeters cubed. These are the surface area and volume for the first sphere, and that gives us a ratio, if we simplify, of approximately 3 to 1. And now we can repeat those calculations for the next sphere. We would have a surface area in the next sphere of 1,256 centimeters cubed, squared rather, and we'd have a volume here of 4,189 centimeters cubed, and this gives us approximately a 1 to 3 ratio. If you think about these two ratios and you think about how diffusion is going to happen, diffusion will be much, much slower in this case. It'll be slower if the ratio is less, and that's because diffusion would have to go all the way across that huge volume of the cell, and it would have very little surface area in order to get into the cell in the first place. We prefer a high ratio for diffusion and the other movements of particles. And this helps to explain why cells are not going to be sort of organism sized. You're not going to be just one giant cell as a human. You're going to have millions and millions of tiny little cells, because you want to keep this surface area to volume ratio as high as possible. When your cells get too big, or you need more cells because you're growing, or because you've injured yourself and you need to repair that, then we have to go to the cell cycle. In order for the cell to copy itself, it needs to first make a copy of the genetic material that's in the cell. And the reason it has to do this is because that genetic material is the code for how that cell is going to function. So if we don't copy the DNA, then we aren't able to function as a new cell. So the way that we'll copy DNA is quite interesting, actually, and there are some fascinating experiments if you are in, a mood for extra reading, to go and see how they discovered this. But if we take a random sequence of DNA and we match up the letter pairs as they would be in the cell, so there's our sequence of DNA, what we're going to do is the cell is actually going to open up that DNA sequence so that the two sides are sort of not connected to each other anymore. And there's a special enzyme that will come along and do that for the cell. Then what's going to happen is new base pairs are going to come and link in, replacing the old pairing that has been opened up. And that'll form a new sugar phosphate backbone and a new section of DNA. You will end up at the end with two sections of DNA, and we call this a semi-conservative model of replication. What that means is that the two new molecules of DNA will be half new and half old. Once the cell has copied its DNA, then we can look at actually sort of dividing the cell. And this happens in 4 steps, and normally we don't refer actually to the steps by name, but I think that it just kind of helps to know the names of these. The first step is called the prophase. If you were looking at a cell in prophase, you'd see that the nuclear membrane might be starting to dissolve ever so slightly, but what would be happening is that your chromosomes would be coming together. So they would have copied the DNA, and instead of the DNA sort of being expanded in the nucleus, the chromosomes are actually forming up as those tight bundles of DNA. Next, we have the metaphase. And in the metaphase, the chromosomes have lined themselves up along the center of the cell. They will be ready to be separated out into the new cells. And that separation is going to happen in the anaphase, which is the third step, where you're going to have the DNA and the chromosomes really being pulled to the new sides of the cell. And finally, in the telophase, you're going to start to reform the nuclear membrane, and you're going to start to pinch in the cell membrane around each of the two new daughter cells. Those are the 4 steps that the cell will go through in order to copy itself, and those 4 steps are referred to as mitosis. It's the process of dividing the cell, and it allows you to repair, for example, a cut. Cell differentiation is the process of becoming specialized, of becoming adapted to the function that the cell is going to have. If you think about yourself and you're about to choose perhaps an A-level course or a college or an apprenticeship, and when you finish that program, you will be adapted to your potential career. You'll have the training and the skills required to do that. With the cells, in some ways it's similar, but in other ways it's much different because they will even change their shape or lose or gain certain parts of the cell in order to become adapted to their function. And they will start out as what we call a stem cell. And this is a cell that can differentiate, so it is undifferentiated at the moment. And that means it can become other cells or other cell types. And we have 2 types of stem cells. The first and the most powerful are the embryonic stem cells. Those are stem cells that come from an embryo. And these cells are really able to become any cell in the body. And I'll remind you that I'm using the C/ abbreviation to represent cell. The embryonic stem cells will differentiate slightly, and they will become what we refer to as adult stem cells. And adult stem cells are similar, but they have a restricted number of cell types that they can become. You might have adult stem cells that can become parts of the blood. So you'd have a red blood cell without its nucleus, and then maybe a white blood cell with its large nucleus ready to fight infections. And then another set of adult stem cells might go on to become skin cells, for example. An adult blood stem cell couldn't magically switch into becoming a skin stem cell. We are currently working on trying to reverse the slight differentiation that has happened for adult stem cells so that we can use them to treat other diseases. But at the moment, the only stem cells that are capable of becoming any cell are embryonic stem cells. There are a few cell types that you need to know their adaptations. That is to say, why are they good at doing the job that they have? So this has a lot to do with the form of the cell, as well as what's in that cell. So if you look, for example, at your sperm cell with its tail, well, the adaptations would be the tail, the fact that there are mitochondria near the tail, and other features such as that. Those allow the sperm cell to do the job of delivering genetic material. A sperm cell, for example, wouldn't be very good at being a fat cell because the fat cell is going to have very little cytoplasm. It's going to have the fat store in the center and then small amounts of cytoplasm. that really what it's doing is storing that fat and being a long-term energy store. Your red blood cell has that sort of characteristic dimple shape, and if you look at it from the side, it's a little bit more obvious. What that's doing is that's increasing the surface area. And red blood cells also have no nucleus, and that allows them to have more space for carrying the oxygen that they're going to carry. Ciliated cells, those cells in your lungs and your trachea that brush the mucus back up into the back of your throat as part of your body's natural defenses, well, their adaptations are those small little cilia on the surface. Another example might be the palisade cell in a plant. So it would have, being a plant cell, would have its cell wall. It will have lots and lots of chloroplasts because a palisade cell is a cell that does photosynthesis. Photosynthesis is the function of this cell. It has to have chloroplasts. I highly recommend that you go and you study your specialized cells and think about, how is this cell good at its job? What shape, what form, what features does it have that makes it adapted to what it needs to do? We've spoken about surface area to volume ratios, and we've said that the human body would be far too large, it would have far too terrible a surface area to volume ratio in order to be able to circulate the things that are all needed to the cells that need them. But diffusion itself, in between all of our small cells, isn't going to be enough either. In order to circulate everything around the body, we have our human circulatory system. And the human circulatory system is a two-loop system. It has one loop that goes to the lungs and one loop that goes to the body. And each of these loops are composed of 3 blood vessels. We have very small blood vessels known as capillaries. We have larger blood vessels with thick muscular walls known as arteries. We have blood vessels with a thinner muscular wall and a very large center known as veins. To go through the features of these, the capillaries have thin walls, and that gives you rapid diffusion. The arteries have thick walls to withstand pressure, and the veins have a large lumen, that center space, in order to allow blood to flow through them freely. They also have valves to prevent backflow. Veins and arteries. Arteries are flowing away from the heart. And veins flow towards the heart. And I always remember this because there's an A in artery and an A in away. Let's go through the 2 loops that form the human circulatory system. So we'll put our heart in the center here, and then I will simplify this loop. So this blood vessel leading away from the heart is going to be the aorta, which is a special artery that leads away from the heart, and it leads into the body capillaries. So there'd be a section of body capillaries all over your body to allow gas exchange and to allow your cells to get the nutrients that they need. And then it will return back to the heart through a blood vessel called the vena cava. If we look at the conditions in this blood, the blood in the aorta is going to be very high in oxygen and very low in carbon dioxide. As it goes through the capillaries, it's going to become lower in oxygen and higher in carbon dioxide because it will give its oxygen to your cells and it will pick up the carbon dioxide that they've used. It'll go then from the body capillaries into the vena cava and back into the heart. From the heart, it's going to go away again through a different blood vessel, and this one is going away from the heart, so it's an artery, and it is going to your lungs. And so everything that has to do with the lungs is pulmonary, so it is the pulmonary artery. And it will come back from the lungs and to the heart. If you think about this, this is blood flowing back to the heart, so this blood vessel must be a vein, and it's coming back from the lungs, so it must be the pulmonary vein. Now, the heart itself is an amazing feat of biology. Our human heart is four-chambered. At the top of the heart, we have two atria, and this is where the blood comes in from the veins. And at the bottom of the heart, we have two pumping chambers, and these are known as the ventricles. Now, if you are looking at a drawing of the heart, you're looking at the heart as if it was sitting in someone's chest. In that heart, the left side of the heart itself is actually on the right side of your drawing. In this heart, these two things here are the left side. The left atrium at the top here, and the left ventricle at the bottom. The other way that you can recognize the left ventricle is that it has a thick muscular wall. And this is because it's pumping blood all around the body, and so it needs quite a lot more strength in order to be able to do that. And then the blood is leaving here, it's passing a heart valve which prevents backflow, and it's going up into the aorta here. On the other side of the heart, we have our right-hand side, so we have blood coming in through here from the vena cava into the right atrium, then into the right ventricle, and then out through the pulmonary artery and out to the lungs. There are some key parts to our blood that we need to know. The first is your platelets, and they are involved in clotting, which means stopping blood flow when there's been a cut. If you have a cut and then eventually the blood stops flowing through that cut, that's because of those platelets doing their clotting work. They won't work on every cut, and certainly not large cuts with rapid flow, but they are there to try and prevent damage to you and blood loss to you. Your red blood cells, we already know they transport oxygen. White blood cells are involved in the immune response. Your blood plasma is the liquid that contains these cells and supports them. And it also contains lots of dissolved nutrients and other materials, and a lot of water, and your body will regulate the amount of water in the plasma in order to regulate your blood pressure. Humans are not the only organism that have transport systems. In plants, we have two types of tissue that do this. We have the xylem and we have the phloem. And the xylem is involved in transporting water towards the tops of the plant from the roots, and the phloem is involved in transporting sugars around the plant. One of my colleagues has a very good metaphor for remembering the characteristics of these two tissues that I quite like, and that is to picture them as stick figures. So first we'll deal with the xylem. Now the xylem is dead, so we'll put little X's on the eyes, and that'll help us to remember that it is the xylem, and it is transporting water, so we'll make the mouth in the shape of water. The arms are going to sort of be down at the sides because it is dead and so therefore not able to raise its arms, and those form arrows indicating the direction of flow from the bottom of the plant or from the roots up to the top of the plant. Whereas the phloem is alive and is celebrating its aliveness, and so that lets you know that you have bidirectional flow. The flow is going from the roots to the leaves, but there's also quite a lot coming from the leaves back down so that the roots can be supplied with the sugar they need because they can't do photosynthesis being under the ground. Unlike humans and other animals, plants do not have a heart to pump their fluids around, and so they need to use something else. And in the plant xylem, we will use transpiration. Transpiration is the process of losing water from a plant's leaves. So we need to look at how the leaves are going to control the loss of water. If you were to zoom in on the underside of a leaf, you would see lots of small little openings around in the base of the leaf that are going to be open to the outside air, and those are going to allow some gas exchange to happen. These openings are called the stomata. They're going to allow carbon dioxide to diffuse in and oxygen and water to diffuse out. That will be the gas exchange that the plants are going to do. The stomata are able to control, they're able to open and close depending on conditions. During the day, when conditions for photosynthesis are good, they are going to open, and at night when conditions are bad, they're going to close, and that's going to help control the movement of water. Now, how do we get water to actually move through the xylem? Well, there are two parts to it. While we're at the leaf, what's going to happen here is the evaporation of water. And that is going to essentially pull the water up from the roots. Down in the roots, we will have osmosis bringing water in, and it will be supported by the active transport of minerals. The plant will create an artificial concentration gradient by pumping minerals using active transport into their root hair cells, and the water will follow those minerals by osmosis. There are a number of different factors that will affect transpiration, and we can look at the rate of transpiration as a function of increasing or decreasing that factor. If we look at light, as you increase the light intensity, more stomata will open and more water will evaporate. That is to say, more transpiration will happen until all the stomata are open, at which point there will not be any effect from having more light intensity because no more stomata can open. The temperature is going to also affect the rate of transpiration because it will allow for more or less water to evaporate. The more you increase the temperature, the more the water is going to be able to evaporate. The next factor will be the air movement. And as you increase the movement of air, you will increase the rate of transpiration. And this is because you'll be increasing the concentration gradient between the inside and the outside of the leaves. But this has less and less effect the more you increase the air movement. Lastly, the humidity, or the amount of water that is present in the air. If you have more water in the air, then less will want to come out of the stomata because the concentration gradient will be less and less important. It will slow down in terms of the amount that is leaving. And how do we measure this? Well, for that we use an instrument called a potometer. And for this instrument, we are going to have an area at one end where we can place a plant, and that plant is going to be able to do some transpiration. So there's my lovely plant drawing. At the stem of the plant, it will be placed in a tube that is entirely filled with water, and it will be sealed up around the stem. So we'll just be looking at the amount of water that is being pulled in at the roots and being transpired from the leaves. That is going to be connected to a tube, which will be connected to a reservoir of extra water, and this is to reset the system. And then the tube will carry along and it'll go past a scale or a ruler so that we can measure some movement here. And then finally it will go into another reservoir at the end so that the water can come in to fill the system. What you're going to do is you're going to make sure that there is an air bubble in this system. And then as the water is being pulled into the plant, it will pull the air bubble along, and you can measure the movement in millimeters per minute. So the rate of transpiration is going to be the number of millimeters divided by the time that it took for the plant to pull the bubble that many millimeters. You can calculate the rate in a number of different conditions, and then you'll be able to discover which conditions have what effect on transpiration. That finishes our notes for B2. I hope that you found these useful. There is a set of revision questions in the description, and the answers to those will be posted next week. As always, if you have any questions, please ask them in the comments, and until then, happy studying.